Welcome to this explainer on the totally fascinating foundational world of genetics and heredity. Have you ever wondered why you might have your mother's eyes, but maybe your grandfather's curly hair? Well, today we're going to cover all of that. We're explaining the hidden mechanics of heredity, complete with images, covering literally all the essential biological building blocks that make you, well, you. Let's jump right in.
Okay, here's our roadmap for today. First, the blueprint of life. Second, the language of genetics. Then we'll look at Mendel and his P's, move on to predicting genetic traits, and finally, we'll wrap up with human sex determination. Section 1. The blueprint of life.
So, it's absolutely crucial we define our terms first. Genetics is, officially, the study of the transmission of body features, both the similarities and the differences, from parents to offspring, along with the strict laws governing how this transmission happens. Think of it like a biological rulebook. It dictates how life perpetuates generation after generation, whether you're a single-celled amoeba, a massive elephant, or, a human being.
Now, you might be wondering who figured all this out. We'll take a look at Gregor Mendel. Born into a peasant family back in 1822, this Austrian monk became what we now recognize as the father of genetics. And get this, he actually failed his teaching certificate exams. No way, right? But his absolute obsession with understanding nature led him right back to his monastery garden. Through brilliant, super patient observation of plants, he discovered the foundational laws of inheritance that we literally still rely on today.
To really grasp Mendel's laws on a modern, microscopic level, we need to zoom way into the nucleus of a cell. When a cell prepares to divide, you can spot these highly condensed, coiled fibers called chromatin, and they have a very specific chemical makeup. They're composed of about 40% DNA, that's deoxyribonucleic acid for the biology buffs out there, and about 60% specialized proteins known as histones. To help us visualize how this incredibly long DNA strand actually fits inside a microscopic cell, our source uses this fantastic analogy. It says, the DNA strand winds around the core of 8 histone molecules, can be imagined like a football around which a long rope is wound. These little complexes are called nucleosomes. Just to put that into perspective, a single human chromosome might have about 1 million of them packed together. It's like a giant puzzle of tightly wound genetic string.
Moving right along to Section 2, the language of genetics.
The most foundational concept we need to get down here is the difference between genotype and phenotype. Genotype is the invisible genetic code. It's the exact combination of genes an organism carries deep in its DNA. Phenotype, on the other hand, is the expressed physical characteristic, what we can actually observe, like your eye color or how tall you are. Basically, the genotype dictates the phenotype. But here's the kicker. 2 organisms that look exactly the same phenotypically might actually be hiding completely different genotypes.
Building on that, we have to talk about alleles. These are alternative forms of a gene that sit at the exact same spot on a chromosome. Now, when a pair of homologous chromosomes carries perfectly similar alleles, like, say, 2 capital T's for pure tallness or 2 lowercase t's for pure dwarfness, we call that genetic condition homozygous. But if the alleles are dissimilar, like 1 capital T and 1 lowercase t mixed together, well, that condition is called heterozygous.
And as you can see in these textbook definitions, we also have dominant and recessive alleles. Think of a dominant allele as the super ruling allele. If it's present at all, it's going to force its trait to be expressed. The recessive allele is kind of the suppressed one. It only gets to show its phenotype if that dominant allele is completely absent, meaning the organism has to be homozygous recessive just to show that specific hidden trait.
Which brings us to Section 3, Mendel and his P's.
Let's connect this science back to us for a second. Heredity is responsible for like begetting like, right? Humans give birth to humans. But it also creates amazing variations. You might share characteristics with your parents, sure, but small differences exist all over the population. We're talking brown versus blue eyes, curly versus straight hair, or even the ability to roll your tongue into a U shape. These are all inherited, alternative forms of a character.
Mendel actually figured out the mechanics of this variation through what's known as the law of segregation. His rule stated that the 2 members of a pair of factors separate during gamete formation. Okay, what does that actually mean? It means that when an organism creates reproductive cells, or gametes, the allele pairs cleanly split up. Each gamete only carries 1 allele for each trait. They don't just blend into a genetic smoothie, they segregate.
And what was his magical test subject for this huge discovery? Pisum sativum, otherwise known as the everyday garden pea. Mendel specifically picked these pea plants because they exhibited very easily observable contrasting traits, things like pure tall versus dwarf stems, or bright yellow versus green seeds. By really carefully crossbreeding these pure plants, he tracked exactly how the traits behaved over multiple generations.
All right, let's jump to Section 4, Predicting Genetic Traits. When biologists want to predict traits, they use different types of crosses. A monohybrid cross is when we breed two organisms while focusing on the alternative traits of just one single character, like just looking at plant height. A dihybrid cross, though, is a bit more complex. That's when we cross parents while keeping an eye on alternative traits of two totally different characters simultaneously, like, say, tracking both seed color and seed shape at the exact same time.
And this perfectly illustrates the math behind it all, the classic 3-to-1 ratio. In a monohybrid cross of heterozygous tall plants in the F2 generation, Mendel consistently found a phenotypic ratio of 3-to-1. So for every 3 tall plants, there was exactly 1 dwarf plant. Phenotypically, what we see is a 3-to-1 split, even though genotypically, those tall plants are actually a hidden mix of pure tall and hybrid tall.
So what happens if we add a second trait into the mix and do a dihybrid cross? Well, the mathematical probability expands. The standard phenotypic ratio in that F2 generation becomes 9-to-3 to 3-to-1. It is incredibly precise. And it totally proves that these different traits are inherited independently of one another, following very strict calculable odds. Today, we actually map out these probabilities visually using a super handy diagram called a Punnett square. Looking at this, you can see how we cross the specific alleles from the parents to predict the exact proportion of genotypes in the offspring. In this example, we're tracking the dominant and recessive alleles for fur color in rabbits to predict the exact odds of the babies being either black or white.
And finally, Section 5, Human Sex Determination.
Now, here's a question humans have literally debated for centuries. Who actually decides if an offspring is a boy or a girl? Does the mother determine it? The father? Is it just completely random? Well, as we've been learning in this explainer, there is a very strict biological rulebook at play here.
Let's break down the human genetic blueprint. We humans have exactly 46 chromosomes in every typical cell. 44 of these are what we call autosomes. They dictate your general body features and are exactly identical in both males and females. But those remaining 2, those are the sex chromosomes. Females have a homozygous pair of X chromosomes, so they're XX. Males, however, have a heterozygous pair, one X chromosome and one Y chromosome, making them XY.
Remember Mendel's law of segregation from earlier? Well, during gamete formation, these chromosome pairs separate. Because a female is XX, 100% of her eggs will carry an X chromosome. But the male gamete, the sperm cell, is different. Since the male is XY, during spermatogenesis, exactly 50% of the sperm produced will carry an X chromosome, and the other 50% will carry a Y chromosome.
So the undeniable biological conclusion here is that the sex of the offspring is entirely determined by the genetic makeup of the sperm. If an X-carrying sperm fertilizes the egg, the result is an XX female. If a Y-carrying sperm fertilizes the egg, you get an XY male. The father's genetic contribution is literally the sole biological deciding factor. It really makes you marvel at the absolute precision of our genetic blueprint, doesn't it? I mean, if everything about us is written in these microscopic coils of DNA, what uncharted recessive traits are hiding in your genotype right now, just waiting for the exact right combination to be expressed in the next generation?